Welcome to part 5 of the chemistry of microbiology. We will be talking about important biological organic molecules. Kindly subscribe to our channel to stay tuned. Important organic macromolecules. Organic molecules contain carbon and hydrogen atoms, and each carbon atom can form 4 covalent bonds with other atoms. The carbon atoms are linked together into branch chains and rings to provide the basic framework of organic molecules. Atoms of other elements, including hydrogen, are covalently bonded to the carbon framework to form a limited number of compounds. Other than carbon and hydrogen, common compounds in organic compounds are oxygen, nitrogen, phosphorus, and sulfur. Others, including iron, copper, molybdenum, magnesium, zinc, and iodine, are important in some compounds. The atoms usually appear in some common arrangements referred to as functional groups. For example, amino functional group, which is found in all amino acids, hydroxyl functional group found in all alcohols, and much are shown in the next slide. There are basic types of organic compounds that are used by all microorganisms. They are termed as macromolecules because they are very large and they include lipids, carbohydrates, proteins, and nucleic acids. Functional groups include hydroxyl functional group that's found in alcohols, monosaccharides, and amino acids. You have ether found in disaccharides and polysaccharides. You have internal carbonyl group that is found in ketone and carbohydrates. You have terminal carbonyl group found in aldehyde. Carboxyl group found in amino acids, proteins, and fatty acids. You have the amino group found in amino acids and proteins. Ether found in fat and wax. Sulfhydride found in amino acids and proteins. Organic phosphates found in phospholipids, nucleotides, and ATPs. Lipids. Lipids are composed of atoms of carbon, hydrogen, and oxygen, but lipids lack the 2:1 ratio between hydrogen and oxygen atoms as in the case for carbohydrates. They are hydrophilic, that is, they are insoluble in water because they are nonpolar molecules. They are rapidly soluble in other nonpolar solvents such as ether, chloroform, and benzene. They are essential to structure and function of the cell membrane because they provide the structure of the cell membranes and some cell walls and functions in energy storage, thus prevent living cells from collapsing in a pool of fluid. Some lipids are used in identifying certain microorganisms, especially bacteria. For example, the cell wall of Mycobacterium tuberculosis, a bacterium that causes tuberculosis, is distinguished from other bacteria by its lipid-rich content. Mycobacterium tuberculosis cell wall contains complex lipids such as waxes and glycolipids that give it distinctive staining characteristics. That's why it's stained through acid-fast staining. The 4 major groups of lipids in the cell include fats, phospholipids, waxes, and steroids, which are discussed in the next slides. Fats. Fats are synthesized in organisms through dehydration synthesis reaction that forms ethers between chain links, fatty acid, and glycerol or an alcohol. A fatty acid consists of a long chain of carbon atoms with associated hydrogen atoms and a carbonyl group at the end of the chain. Fatty acids can be saturated or unsaturated. A saturated fatty acid contains the maximum number of hydrogen atoms it can accommodate, that is, it is saturated with hydrogen. An unsaturated fatty acid has lost at least 2 hydrogen atoms and contains bonds between 2 carbon atoms. A fat is formed when a molecule of glycerol combines with 1 to 3 fatty acids. The number of fatty acid molecules determines whether the fatty acid molecule is monoglycerol, that means it has one fatty acid, diglyceride when it has two fatty acids, and triglycerides when it has three fatty acids, which is the maximum number of fatty acids it can contain. In the reaction, 1 to 3 molecules of water are formed. That's dehydration, depending on the number of fatty acid molecules reacting. The chemical bond formed when the water molecule is removed is called an ether linkage. In the reverse reaction, hydrolysis, a fat molecule is broken down into its component fatty acids and glycerol molecules. If several double bonds exist in even one fatty acid of the fat molecule is called polyunsaturated fatty acid. Saturated fats, such as those found in animals, are usually solid at room temperature because their fatty acids are relatively straight and can be packed closely together. An example is the one shown where the glycerol reacts with fatty acid to form a carboxyl group to form water molecules. Fats. Unsaturated fats remain liquid at room temperature because their fatty acids are bent at every double bond and cannot be packed tightly. If the hydrogen atoms on either sides of the double bond is oleic acid are on the same side of the saturated fatty acid, the fatty acid is called a cis fatty acid, while if on opposite sides of the double bond, the acid is called a trans fatty acid. An example showed in the previous page is one of the cis fatty acid. Most plants fats are unsaturated or polyunsaturated. Fats contain a large amount of energy stored in their carbon covalent bonds. Thus, its major role in organism is storage of energy. They are catabolized to provide energy for movement, synthesis, and transport. Phospholipids. They are made up of glycerol, two fatty acids, and a phosphate group bonded to one of the several organic groups. Phospholipids are the lipids that build membranes. They are essential to a cell's survival. Phospholipids have polar as well as nonpolar regions. When placed in water, they twist themselves in such a way that all polar, that's hydrophilic portions consisting of a phosphate group and a glycerol orient themselves towards the polar water molecules, with which they form hydrogen bonds that hydrophilic, meaning water. Loving. This forms the basic structure of plasma membrane. On the other hand, all nonpolar, that's hydrophobic, parts of the phospholipids consisting of fatty acids make contact only with the nonpolar portions of the neighboring water molecules. Hydrophobic means water-fearing. This characteristic behavior makes phospholipids particularly suitable for their role as a major component of the membranes that enclose cells. Phospholipids enable the membranes to act as barriers that separate the content of the cell from the water-based environment in which it lives. Example of phospholipids is shown where we have organic uh, molecule, that's glycerol, reacted with a phosphate group, unsaturated and one unsaturated fatty acids. And the other side you'll see is showing the cell membrane structures. Waxes. Waxes contain a long chain of fatty acids linked covalently to a long chain of alcohol by an ether bond. They are completely insoluble in water. Some microorganisms like Microbacterium tuberculosis, the bacteria that causes tuberculosis, are saturated by a waxy cell wall, making them resistant to desiccation. Some marine microorganisms use wax as energy storage molecules instead of fatty acids. Some complex lipids are useful in identifying certain bacteria. For example, the cell wall of Mycobacterium tuberculosis is distinguished by its lipid-rich component. The cell wall contains complex lipids such as waxes and glycolipids that give the bacterium distinctive staining characteristics. Thus, only as its fasting used in the identification of steroids. They consist of four rings, each containing 5 or 6 carbon atoms, which are attached to various side chains and functional groups of one another. They play many roles in metabolism. Some act as hormones, others like cholesterol, which is another form of steroid, as an essential part of phospholipid bilayer membrane surrounding an animal cell. Cell membranes of fungi, plants, and mycoplasma have similar sterol. That's another steroid. Sterol has a functional group hydroxyl, that's the hydroxyl group, which interferes with the tight packing of the fatty acid chains of phospholipids, thus making the membrane and to keep fluidy and flexible at low temperatures. That's why without steroids such as cholesterol, the membranes of cells would become stiff and if inflexible in the cold. An example of is cholesterol. The structural formula is as shown in the image. Thank you for viewing. Kindly subscribe to our channel to stay tuned.